The Earth Biogenome Project is an international network of networks that has a common goal of sequencing all approximately 1.8 million named eukaryotic species. It is the coordinating structure for all these individual projects that are located around the world, providing the governance, standards, and ethical framework for the project to succeed. The international network of networks consists of 60 laboratories around the world who are coordinating together on standards and producing these genomes at scale. BGI has been the focal sequencing center for many of these other projects around China before they developed their own sequencing capacity. BGI has been doing the sequence for many of these projects, like the bird, the B10K project, the ants project, and several of the other projects. So BGI has been contributing large numbers of sequenced genomes toward the goals that we have been setting for the project. Why do we want to do the Earth Biogenome Project? First, we are losing biodiversity on the planet at an alarming rate. Species are disappearing, and it is projected that if the current rates continue, by the end of the century, we may lose half of the species on the planet. And nobody knows the tipping point when we can reach the point where ecosystems on the planet will collapse. If ecosystems collapse, that could affect our ability as a species to survive. The critical ecosystems for which we depend on for food and for water and for clean air. And so we must make every effort to preserve the organisms and understand their genomes in order for us to properly conserve and manage biodiversity into the future. Number two, bioeconomy. New medicines from nature. Today, 80% of the medicines that we take as humans have their origin in nature, in natural products. In China, you've been a leader of this for 5,000 years with traditional Chinese medicine, and the rest of the world is just catching on. So being able to understand those organisms, and because only a small fraction of them have had their genome sequenced. So very important for development of new pharmaceuticals for plant and animal health, and also for agriculture. We need to identify wild relatives of some of the major species that we use as food crops and for animals, because through the selective breeding processes that we've been using for the last 75 years for plants and animals, we are restricting their genetic variation. And we need often to bring in alleles, variants of genes from wild relatives of species used for agriculture to allow us to bring resistance to specific diseases or new diseases that can emerge, new pandemics that may emerge in plant and animal populations. The beautiful thing that has happened is that technology has advanced so rapidly that we can now sequence genomes and annotate them, find their genes at a fraction of the cost that it took when the human genome was sequenced. So the next 20 years is continued improvements. Costs will come down further, and the accuracy and length of these assemblies is going to just improve over time. The second big advance, as we're seeing, is probably going to be AI. On top of all these tremendous advances that we've seen in biocomputing and bioinformatics, now we're going to have a massive training set of genomes to train the large language models so that we can move into an era of AI-driven discovery and AI-driven applications that will come from this massive database of sequenced eukaryotic genomes. Within 2 or 3 years, we're going to see tremendous advances that are going to derive, practical advances that will derive from the sequencing of these genomes. Return on the investment to society, that people are going to benefit with better products for agriculture, newer medicines. We're going to be able to preserve not just species, but entire ecosystems that are threatened on the planet. And I hope to see that in my lifetime.